Bismillahirrahmanirrahim, and welcome back to this next video. And this is the second video in the series of videos on the bioinformatics. In the previous video, I've told you about the definition of bioinformatics, that bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data, in particular when the data set they are large and complex. And I've told you about the in vivo studies, about the in vitro studies, and then about the in silico studies, which are actually the bioinformatics studies. Then we talked about some of the applications of bioinformatics, that the centrality of the bioinformatics to the cutting edge developments in molecular biology. So people from many different fields, they have been stumbling across the term in a variety of different contexts, like the biology, medical or computer science students, a professional from a pharmaceutical industry, a lawyer or a policeman about the DNA testing, consumer which is concerned about the genetically modified organisms, and even a Nasdaq investor who is interested in a startup company. Now in this particular video, I want to focus on how this thing started, how the bioinformatics, the field of the bioinformatics that started. And it actually started from analyzing the protein sequences. So in the early days of biochemistry, the biochemists, they were mostly focusing on finding ways to have a better way to represent proteins in terms of formula that would explain their biological or even their nutritional properties. So that was the main focus of the biochemist in the early era of the bioinformatics. Now, after some time, the biochemists realized that proteins, they are very huge molecules. Technically, we call them as the macromolecules. And these proteins, they can be made of from their building blocks, which are the amino acids. And a single protein usually contain about 100 to 500 amino acids. And these amino acids, they are picked out from a selection of 20 amino acids. So simply you can say that they realized that the proteins, they are made up of the amino acids, and these amino acids, they are picked from a selection of the 20 amino acids. Now this particular table is going to show you three things: the complete name of the amino acid, their 3-letter code, and their 1-letter code. So there are usually 20 amino acids. They are used in the synthesis of the proteins, and these are represented by sometime 1-letter code, sometime by 3-letter code, and sometime by their full name. So the for example, the first amino acid, that could be the alanine. So this is the full name of the amino acid, the ALA, that is a 3-letter code, and this single A, it is actually representing a 1-letter code for the alanine. Same is true for all of the amino acids, like the arginine, the asparagine, the aspartic acid, the cysteine, the glutamine, the glutamic acid. There are 3 and the 1-letter code that has been mentioned against their name. These are the other amino acids, the glycine, the histidine, the isoleucine, and then the leucine, the lysine, methionine, phenylalanine, the protein, serine, threonine, tryptophan, tyrosine, and valine. So this is the 20 set of the amino acids from which the proteins they are synthesized. Now from where these names and these 3 and a single letter code comes, these full name, these 3-letter code, and these 1-letter code for these amino acids, they're actually designated by the International Union of Pure and Applied Chemistry Committee, which is for sure known as the IUPAC. Now, the biochemists, after some time, they recognized that a given type of a protein, for example, if you talk about the insulin or the myoglobin, they always contain precisely the same number of total amino acids. Sometimes this term residue is also used for the amino acids. So they realized that a single kind of the protein, a given type of a protein, they contain precisely the same number of total amino acids in the same proportion. At which time you collect the sample, at which time you collect the insulin from an individual, it will also always contain the same number of total amino acids. Say for example, so a better formula for a protein like the insulin. So insulin, for example, will always contain 30 glycines. It will always contain like 44 alanines, the 5 tyrosines, or the 14 glutamines. What I mean to say is that a given type of the protein all of the time will contain precisely the same number of the total amino acids. Now, finally, the biochemists they discovered that these amino acids are linked together as a chain, and that the true identity of the protein is derived not only from its composition but also from the precise order of its constituent amino acid, which we technically call is the primary structure. What this means is that not only the total number of amino acid is important, but also the sequence of the amino acid that is important to determine the biological function for a given protein. Now, the first amino acid sequence of a protein that was determined in 1951, and that was discovered by that was the protein insulin, whose sequence was determined in 1951. So we will continue this discussion, the genesis of the bioinformatics, in the next video.